Okay, so let's begin. At this seminar we have a speaker, and I think you must have met him, this cheerful fellow who is always around the building A31. His name is Ortiz, and he's a postdoc in my lab from the April this year, right? From April. So to give you a short overview, who is he? So David has studied biology at the Universidad de La Habana in Cuba. And then he did his PhD in Mexico, actually at the Universidad Nacional Autónoma de Mexico, right, in Mexico City. In the meantime, however, he has traveled a bit. So first of all, he was back in Cuba, where he worked at the Institute of Ecology and Systematics. Then he was also appointed at the University of Havana. And then he moved to Spain, where he stayed both at the University in Madrid and in Barcelona. And he's finished his PhD on the topic of the phylogenetics and systematics of theraphosid spiders, so-called tarantulas. Here, he's actually continuing in his topic, so he's working on a phylogeny of a different spiders, however. And that's much smaller than the tarantulas typically, and as you will see actually in this talk. So, but before he begins, I'll need to send you the list that you have, the sheets that you have to sign. There are two different sheets, so please do remember to sign both, and then please also do not forget to vote for the best poll of this semester. So David, here you go.
Stano, thank you for the introduction and for giving me the opportunity, and good evening to all, and thank you for being here. Today I'm going to talk about my research back in Mexico City for my PhD, and now my postdoctoral research here in the Masarykova University. The title of my presentation is Different Origins but a Similar Fate, Molecules Uncover High Diversification and Slow Morphological Change in Mexican tarantulas and Iberian eating spiders. So, since the advent of the theory of evolution, and since we discovered that all organisms in this planet they share a common origin, so phylogenetic inference have become a central issue in the study of biogeography, for example, trait evolution and, of course, systematics. Within systematics, species delimitation is one of the key issues because it's all around, it's work for everything. For example, it's key for conservation biology, it's very important even for public health, and also it's very important to assess our own history. These delimitations have been traditionally estimated using morphological features. Morphological species delimitation have several advantages. For example, it is inexpensive to be employed. You don't need a lot of equipment. It can also make use of a huge bank of biological collections that we have stored for several hundred years in museums. It can also get use of fossil material. And species identifications and delimitations can be performed even for non-specialists in the field. However, species delimitation purely based on morphology have several disadvantages. For example, it's difficult to assess the relationship below species level. This is because we normally cluster individuals as species using morphology because of the lack of differences, instead of the presence of shared features. Morphology is really strongly influenced by selection pressures. For example, you can have very successful morphological patterns that remain very stable for sometimes millions of years, whereas there can be quick diversifications in other situations. And within the same species, you can have, for example, castes in the social insects. You have different morphologies, easy to differentiate, but they don't represent different. So if we don't know about this, we can fail at species delimitation based on morphology. Also, morphology-based species delimitation have been criticized for being too dependent on the opinion of particular researchers. So the delimitations are not performed on a mathematical or on a quantitative background, but we normally use qualitative characters, and we decide if we are talking about the different species or not, and that differs between researchers many times. That's why some species go to be synonyms, they are revalidated, go back, and there's some instability about this. And we are also very dependent on specific sexes or life stages. For example, in many species, only adult males or adult females, or even in some other juveniles, are those that bring information at species level. So we can, in species that are hard to find, we can go to the field, spend a lot of money, and if we don't find the right individuals, we cannot do it. Fortunately, since like 30 years or so, molecular information are bringing new lights into the species delimitation problem. So molecular information bring information below the species level. So we are for the first time able to assess the using different and modern species concept, for example, biological species concept, genotypic cluster, phylogenetic, and some others. Initially, the species delimitation based on molecular data were only based on DNA barcodes, which is the use of one or a few linked molecular markers to establish the species identity and to perform species delimitation. This is, has many advantages because it's normally easy to implement. The molecular markers, normally we choose those that are highly informative, but at the same time, the fact that it is based on a really tiny fragment of the molecular information can give a strong bias in the results. So they are really dependent on the information present in those particular molecular markers. In the last years, several multi-locus methods of species delimitation have been developed. For the first time, we are able to use like sometimes hundreds or thousands of molecular markers at the same time to the species delimitation problem. These analyses tend to be highly reliable because we are using a huge amount of information that is not so biased as before. But in this point, we are not, I mean, it's still very expensive to get all this data. It requires a lot of work, and also the analyses are time and resources consuming. So we are not in a minute in which we can perform like multi-locus species delimitation really easily, and it's not a reality yet. Especially for neglected organisms, it's really to employ this because we don't have normally a lot of funding to perform this expensive analysis, and basically multi-locus methods have been employed in really the groups that get the most of the funding. That sometimes they are not the most diverse, or many times. So my work basically is about systematics and evolutionary biology, and I'm interested in how to use molecular information to assess phylogenetic relationships, how past events shape present distributions, and to assess morphological, ecological, behavioral diversification, etc. And also to employ new approaches to determine species boundaries and perform species identification. So, but we cannot use everything. So I use spiders, which is a very interesting group to conduct evolutionary studies, because spiders are very successful animals. So far we have described more than 40,000 species worldwide. It's a group that have really diversified a lot. They have generally low dispersal capabilities. That makes them very prone to suffer from evolutionary processes locally. So there's a big space for conducting different studies with them. And they have suffered a high morphological and ecological diversification. So let's go straight to my work during my PhD was with tarantulas, or theraphosid spiders, which is, they comprise around 1,000 species distributed in all the tropical and subtropical regions of the world. Despite there are 140 genera described and 9 to 13 subfamilies, about half of the diversity of all tarantulas is located in the subfamily Theraphosinae, which is endemic to the neotropics. The systematics of tarantulas or Theraphosid spiders bring a lot of problems. For example, they have a low morphological and ecological diversification. We can see here two species of tarantulas. They are from different subfamilies, but they share the same morphological pattern despite the difference in color that you clearly see. But if we go to other families of spiders, we can see, for example, in Salticidae, the morphological pattern is completely different between different species. So you can see there a high diversification. So we have in Theraphosidae a strong morphological stasis, and that give a lot of problems for species delimitation, for phylogenetic inference based on morphology, etc. The sexual features, which are normally very important for delimitation in species delimitation in spiders, they have low complexity when compared with other spider groups in tarantulas. And if we add that there's a relatively high intraspecific variation, for example, here you can see different spermatheca, which are the sexual structures of females of a single species. And not only a single species, these are all individuals from the same locality. So we have a low complexity and we also have a high intraspecific variation. So that's the perfect mix for a nightmare group to study. And so morphology-based species delimitation in tarantulas are ambiguous. Morphological phylogenies with low resolution and support. They have been done in about the last 20 years, but morphology-based phylogenies have always the same problem: low branch support, low resolution. So they haven't been really able to assess the phylogeny of the group. So the current status, the intrafamiliar relationships of tarantulas in general are very poorly known. And since the year 2000, we have an additional problem. People are describing genera like very freely. So since 2000, we have 23 new genera with only one or two species described. So the taxonomy of the group is really becoming a big mess. We need a phylogeny to give order to the taxonomy of the group. So in the last years, several researchers started to use molecular information to address the taxonomic problems in tarantulas. Most of these papers, or all of these papers, have been addressed to species delimitation, not to the phylogeny. And in the most striking of the results, Hamilton et al. In 2016 synonymized 33 of the 55 previously recognized species of Aphonopelma from the United States and described 14 new species. So there's a big difference between the delimitations obtained from morphological data and from molecular data in tarantulas. So the objectives of my thesis were the systematic revision of Bonetina, which is a genus, small genus endemic to Mexico. They were only known from 4 localities before we started working with it, but we had plenty of material from in biological collections, so we knew that there were more species to be described, and integrate morphological and molecular information at all levels of the systematic work, including species delimitation that will address first, species diagnosis and description, the purely taxonomic work, and phylogenetic inference and trait reconstruction. I will address these different topics in segments. So first, it was the species delimitation. We published that paper in 2016. And the objectives of this paper were to determine the congruence of species delimitation between morphology and the quick evolving mitochondrial marker CO1 and quick evolving nuclear ITS in Bonetina. Most species delimitations based on molecular data and in tarantulas have been performed using CO1, which is a good marker. It's the most widely used for species delimitation in animals in general, but we talked about before about the problems of using only one marker. So we wanted to know if using a different marker additionally could shed new light on the delimitations in the group. So compare several methods of molecular species delimitation and obtain an iterative hypothesis of the species richness and identity within Bonetina. Also, we wanted to determine the influence of the alignment method and of coding gaps as characters on phylogenetic inference and species delimitation with ITS. The thing is that ITS is a non-protein coding gene, therefore it evolves in the forms of substitutions, but also very commonly as insertions and deletions. So when you have a big divergence in your group, you are going to have a lot of gaps and the alignment is ambiguous. So we wanted to address if there was a lot of ambiguity in the alignments and how this ambiguity impacted the delimitations and the phylogenetic reconstruction. So we went through the whole distribution range of the genus, collected specimens, addressed morphology, and sequenced CO1 and ITS1, and performed the alignment using 3 different methods. One simple method using Clustal W2 is not a really method, it's especially adequate for easy sequences with not a big change, not big gaps. Then we use two stronger methods like MAFT and PRANC. MAFT is an iterative method, and PRANC is a Phylogeny dependent alignment method. Also, we address how the coding gaps as characters in the case of ITS1 influence the topologies obtained in the phylogenies and also the species delimitation. So, for performing the species delimitation based on morphology, we use the traditional thing, like mainly, we assess all the characters we found in our animals, but we also especially focus on the sexual structures that tend to be more variable at species level or at all levels. And for DNA, we employ single locus methods based on genetic distance, ABGD, and the hard gap barcoding. And phylogeny-based methods, we have a stronger theoretical background using GMYC, TTP, and BPP. So all these analyses were performed with CO1 and ITS1 separately. And then at the end, we employ BPP, a multi-locus method, to perform species delimitation based on both markers at the same time. Well, the results, we had 76 specimens sequenced from 38 localities. Like we had 4 localities before we started the project, so most of the localities were new. And the alignment of CO1, as expected, was unambiguous, no indels. The maximum p-distance within our matrix of CO1 was 13.4% between sequences. That's a pretty deep divergence within a single genus. And in the case of ITS1, you can see here all these gray squares, they are indels. So the alignment was really highly ambiguous. Different alignment methods gave alignments completely different to each other. The length of the alignments varied between 652 and 706 base pairs between the 3 different methods. And the distances between the alignments between 8.8 and 10.2, which is pretty high. Coding gaps as characters, we obtained 94 to 116 new characters only by coding gaps in ITS1. So, yeah, it was pretty messy. And the maximum distance within the matrix was also very high, 13.8%. So, let's go to the results. This is the summary tree only from the ITS1 marker. The blue branches represent branches recovered across all analyses with high support. High support meaning more than 80% of bootstrap in the RAXML analysis, maximum likelihood, and more than 95% of posterior probability in Bayesian analysis. The squares here represent the first row is the alignments with MAFT, PRANC, and Clustal, and the columns represent the DNA matrix only and the DNA plus the gaps coders as characters matrix. So we find that despite the huge difference in the alignments, the topology obtained from them were very similar. And this is really what we are interested in. I mean, the alignments were different, that's true, there's a lot of ambiguity, but if the trees are the same, we are happy. So, by coding gaps as characters, it barely influenced the topology of the tree. We got about the same, but the branch support was improved between 2 and 10%, which is good. In case of species delimitation, here you can see the species tree and the results. These columns are the result by the different methods of delimitation. And here is a summary. This is morphology, CO1, ITS1, and the combined ITS plus CO1 results. As you can see, there's a lot of variation between the results obtained from the different lines of evidence and from the different methods employed in the analysis. For example, morphology, we got 9 species in our database. With CO1, we estimated that we had 14 species and a variation between 12 and 18 between different methods. In case of ITS1, 10 species as like the strongest supported solution, but a huge variation between 1 and 20, so it was absolutely not helpful. And the multilocus delimitation varied between 15 and 17 species. I mean, multilocus, we are talking about only 2 genes. We had only CO1 and ITS1. We're not talking about big information. The integrative solution for us was like we had 12 strongly supported species across the analysis, and 7 of these species were new. Okay, the next thing is about the taxonomy work, how we translate to the taxonomy the result we obtain. I'm not going to focus on this here because this is mostly relevant to the people who work on the group, but I can tell you that when in this paper we use more animals that we previously used for the species delimitation. We had to rerun the whole thing again. We got 17 solidly supported species in Bonedina, 12 were newly described during our work, and we offer molecular diagnosis of species based on genetic distances, and also we found autapomorphism for most of the species. Here you can see a little bit the diversity within the group. This is the distribution. The distribution is not very big, it's about 900 kilometers in the most isolated places, but we are talking about, this is central and southern Mexico. It's an area with a huge biodiversity where different biographic areas converge, so it's not surprising that we found such a big diversity in Bonedina. And talking about the morphologies, you see that this for tarantulas is a big difference. I mean, this is a big difference between species, and you can see now Bonedina is one of the most heterogeneous, morphologically speaking, genera in Theraphosidae. But what about the phylogeny? We published the phylogeny paper in 2018, this year, and the objectives of this work were to evaluate the monophyly of Bonedina, determine its phylogenetic position and relationships, estimate the temporary framework of the evolution of the group. We have no idea how all these Theraphosidae or tarantulas, we have no idea of when the different groups diverged. We only have 2 fossils, both of them from the Miocene, one from Mexico and one from Dominican Republic. We are talking about between 18 and 20 million years. So the information on the evolution in a temporary framework of tarantulas is absolutely inexistent. We wanted to perform an ancestral state reconstruction to assess the morphological homoplasy in the group, because the morphological characters at the end are going to be those that we normally use to define bigger groups, like superspecific or suprageneric groups in Theraphosidae. So we wanted to assess if the morphological homoplasy was really that high or not, and determine what characters were good to define groups, determine the probable ancestral areas of distribution of the main groups, and for this we use the 17 Bonedina species, 15 other Theraphosidae genera, because we had no idea of the relationships of Bonedina with other Theraphosids, so we tried to choose a big number of outgroups that represent groups that were probably closely related or that lived in the same geographic areas, and a number of also from the determined lineages that there are really new genera that have to be described. Okay, so what kind of information did we use? We used the molecular markers we use for species delimitation, CO1 and ITS1, but in this case, as is for phylogeny reconstruction, we added 4 new markers. They are nuclear markers, they are unlinked between them, and although the variation is much lower than that we obtain with CO1 or ITS1, they are good to support the deepest branches of the tree. The phylogenetic inference was made in maximum likelihood and Bayesian background or whatever. RAXML was employed for the maximum likelihood reconstruction, and Mr. Bayes was used for Bayesian analysis, and BEAST was used to estimate the divergence times between the groups. So we also built a morphological matrix with characters, a new matrix, to assess how was the morphological evolution in Theraphosidae, or in the group we explored. We found a number of new characters using scanning electron microscope that were not targeted before, etc. And ancestral states were estimated in a maximum likelihood, I also forgot the word again, with mesquite, and Winclara was used to do it in a parsimony environment. Maximum likelihood has theoretically better resolution to estimate ancestral reconstruction. However, parsimony-based reconstructions are good because you can see all characters in a matrix at the same time, so you can assess the validity, or you can determine well how big is the homoplasy in the group. And the ancestral areas were estimated with RASP. So we have here the summary tree that we obtained. We use 88 specimens of 48 species, including the 17 Bonetina species. Again, the blue branches represent branches recovered across all analyses, meaning RAXML, Mr. Bayes, and BEAST, with high support, more than 80% in Bootstrap for RAXML, and more than 95% for the Bayesian analysis. So you can see that the topologies are very similar, and they have a pretty high support. There are a few branches. You see here the red spots, dots. They are not recovered in all three analyses, but this is really a minority when you compare with the whole topology of the tree. And some of the results were that the North American and Central American tarantulas with type 3 Ortiz seta, they form a monophyletic group. Bonetina, it's here and here. It's polyphyletic because Bonetina justaventricola is not really has a different origin. It's not really a Bonetina. But if we exclude that species, Bonetina starts to be a monophyletic group. And Bonetina is sister to those two species. They are undescribed, that they lack both morphological features that diagnose Bonetina. Therefore, we are talking about a different genus, undescribed. Within Bonetina, there are three groups of species. If we go to the chronogram to see the times of divergence between the different groups, we determined that the most recent common ancestor of all Theraphosids used in our analysis, this includes all this, the neotropical taxa plus Ichnocolus, which is the L group that we use to root the tree. It's from southern Spain. They diverge as a medium value as about 92 million years. That makes sense biographically because South America and Africa split about 110 million years. So this 92 is the medium value, but the divergence, the confidence intervals include the time in which South America and Africa split. The clade with Central American, North American tarantulas with Seta type 3 arose by the Middle Oligocene about 30 million years. The most recent common ancestor of Bonetina is 17 million years, so the genus is pretty old. And the most recent common ancestors of the groups within Bonetina, Aviae, 14 million years, Papalotensis 10, and Cyanifemur 9.4. So we're talking about a genus that has a pretty deep evolutionary history. And there's a remarkable sexual diversification, especially in the male sexual characters. As you can see here, for example, this one is completely different pattern, up to until the Middle Miocene. So after the Middle Miocene, all diversification gave rise to species that they have the same morphological pattern in the pedipalpal bulbs. If we go to the ancestral state reconstruction, here you can see the black squares are the sign apomorphism. I mean, characters that define groups, they are not ambiguous. And the white squares, they are homoplasys, meaning characters that they appear in different times in the evolution of the group. As you can see, most of the branches or either have no characters defining them or they are based on homoplasys. That supports the idea that the group have a really conflictive morphology because the same features arise once and again in the evolution. So that makes it very hard to define groups based on morphology in Theraphosidae. And finally, if we go to the reconstruction of ancestral areas using RASP, we can see that most of the ancestors of all neotropical tarantulas have an origin in South America. I mean, after the split from the Old World, during like 40 million years, between the 65 and 30 million years, all tarantulas from the neotropics, they lived in South America, apparently. So South America at that time was an isolated continent. It was not together North America, there were not the Isthmus of Panama yet. So apparently most of the early diversification in the group in the neotropics occurred in South America. But there were two different events of invasion of North and Central America. One of them for the species with type 3 orticating seta on the abdomen. This happened about 35 million years in a time in which still South America and North America were not connected, but there were some islands in the middle, so it is possible that the tarantulas were able to disperse through island hopping to North America. This also happened not in many groups, but in some groups. Most of the biological connection arose between North and Central America after the rising of the Isthmus of Panama, but still there are many groups that did it before. And there was a second invasion event during about 15 million years ago of the species with type 1 orticating hair in the abdomen. So most of the diversification occurred in South America. So as a result of this project, we were able to answer many questions we want, but always you get new questions after this. So for example, will other groups of poor dispersal spiders show similar remarkable genetic and species diversity hidden behind a conserved morphology as in Theraphosids? What was the influence of the fragmented nature of our sampling on the species delimitation results? We know that molecular delimitation results and also morphological delimitation results, if you don't accomplish the whole diversity in morphologies and genetic diversity, you are somehow like creating boundaries where there possibly could be no boundary. So it's important to have an as complete as possible representation of the whole diversity to perform delimitation properly. So in the case of Bonedina, we didn't have this sampling. I mean, 12 out of the 17 species that arose from our project in Bonedina were living only in one locality, because I mean the distribution area was pretty big and we didn't have the time to go every number of kilometers and do a more thorough sampling. Also, the Bonedina were not easy to find. There was a time in which we did a trip of 10 years and we didn't find a single specimen, so it wasn't nice. And another of the questions would be, would the species delimitation results based on one or a few markers? Because we saw that there's a strong variation of the results when you use different markers and different species delimitation methods based on one marker. So would these results be very different from those calculated from hundreds of markers, I mean like genomic-wide species delimitation? So that's why we have a new project now here in the university, and we are addressing these and other questions that we had. In this case, we're working with Sodarium. It's a completely different group of spiders. They are small, but they are very diverse. Mostly they live across the Mediterranean region, but some species go to Central Asia. So far, there are 160 species described. And there's a low morphological diversification also in Sodarium. The dispersal capabilities of the group are supposed to be low. For example, they live in these structures under stones. We call them igloos. And there are many spiders who, when they are juveniles, small spiderlings, they create a small thread and they can transport themselves through the air. And this way they can invade different places. But the Sodarium don't do it. So that is not a possibility for them. Also, Sodarium spiders feed exclusively on ants, but not only on ants. They depend sometimes or many times in specific ant species. So there's strong diversification and specificity towards the prey that is likely to produce also a barrier for dispersion or dispersal. This species delimitation, of course, as always, is mostly based on morphology. And well, the objective of this project were to test the reliability of morphology and DNA barcodes for species delimitation in Sodarium styliferum, a species that is widely distributed across the Iberian Peninsula. To test the hypothesis of the existence of morphologically cryptic species in the group, in cryptic lineages determine if the few lineages have diversified in biology, specifically in their interaction with prey. Why we think about there could be cryptic species? The thing is that most Sodarium species live in really small places. Like the distribution is really small, but there are a number of species that they live across bigger areas. So we wanted to know if these species really live there, or we are talking about groups that are morphologically very conserved, but they are really more diversified. So we also want to study the biogeography of Sodarium styliferum in a historical context and determine what were the drivers of its expansion across the Iberian Peninsula. And finally, as a side project, infer the phylogeny of the whole genus Sodarium using as many species as possible, and this way determine the time frame of the evolution of the group, its historical biogeography, and aspects of its morphological, behavioral, and physiological diversification. Well, why Sodarium styliferum? Well, the thing is that, as I mentioned before, the distribution of the species is pretty wide when you compare with other Sodarium species. They are highly abundant in the habitats where they live, so we were pretty sure that we were going to be able to collect these species, and if you don't have specimens, you don't have a project. And there's also a taxonomic conflict with other species. There's Sodarium extraneum that sometimes is treated as a synonym of Sodarium styliferum, and sometimes they are treated as different species. So we came through the distribution range of the species in spring of this year, made a long field trip of 3 weeks, covering the whole distribution of the species. We try to sample localities every 50 kilometers to try to avoid the artificial exaggeration of the genetic divergence, and to try to avoid singletons in case of the deep genetic structure. So we tried to collect several specimens per species per locality, and we collected all other Sodarium species we were able to find for the phylogeny project. And also to assess if Sodarium styliferum was a monophyletic group at least, because we didn't know that. So, 2 individuals per locality were sequenced using CO1, the animal barcoding marker, and 1 individual per locality is going to be sequenced using ddRat, which is a variation of RAT-seq, a methodology that based on animals that you don't have any previous information, you can sequence from them like thousands of loci at the same time at a relatively low cost. So far it's like 550 Czech crowns per sample if you sequence a whole lane. So it's more affordable than other genomic approaches. Species delimitation, again, using morphology, and for the single locus we chose some of the methods that have given us better results in the past, using hard gap barcoding, GMYC, and PTP. And for the multi-locus method, we are going to use like the big information here using BPP that is supposed to give highly reliable results. And this way, we want to compare the methods that that have low cost and effort, the morphology and the DNA barcoding, against the high reliable method of delimitation using big information, which is not so easy to implement so far. So that was one of the... In case of phylogeography, we want to determine the dispersal capabilities of the species, determine if there's isolation by distance, if there's evidence of genetic flow between populations of the so-called Sodarium styliferum, determine how geological and climatic past events have shaped the distribution. And this is a work in progress, so we are going to employ different methods based on the data that we get. We don't have this data yet. So, when we have the species properly delimited and we know how many potential species are within Sodarium styliferum, we want to do a shorter field trip and collect alive specimens from species previously delimited and perform experiments to determine if there's reproductive isolation between these different species, diversification in prey preference, venom composition, efficiency, etc. For this, we are going to follow the methodologies that have been employed in several works in our lab in the past, because in our lab we normally do this kind of work. And for the phylogeny of the whole Sodarium, we count with about 80 species, counting Sodarium and outgroups. There are a total of 160 species of Sodarium, but we have a representation of the whole distribution range of the genus, so this analysis should give us a very good idea of how the relationship between the main groups are. And we are going to sequence, in this case, using another methodology that it's a derivative from RAD-seq. It's called HiRAD. In this case, HiRAD, which is more expensive, but allows us to sequence specimens from museum, because most of the material we have from all these species come from museums. So in this case, we're going to get a lot of information, but we are going to be able to use museum material, which is great. And the phylogeny and divergence dating will be performed also using RAXML, Astral2, and BEAST for the divergence time estimation. Finally, when we have the tree, the phylogenetic tree, we will be able to assess the taxonomy of the group, determine what morphological features are the best to define groups in Sodarium, assess the morphological evolution, see if some characters, some traits have promoted diversification within the group, etc. But this will be after we have the phylogeny, and determine the historical biography of the group, determine the drivers of the diversification of Sodarium across the distribution range, when this occurred, and how the geological events that have happened in the area influence the distribution and diversity of Sodarium. Well, we already have preliminary results. From this trip, we were able to collect Sodarium styliferum in 74 localities out of 103, which is, I was very happy about that. But I wasn't very happy because they really like ugly places. I mean, we were traveling across very nice places, but the Sodarium styliferum, they really like the worst spots in the area. So they really like human populations and dumps, etc. So if you want Sodarium, just go to the styliferum, just go to the worst spot. So the CO1 diversity, we already have the CO1 data, show a huge diversification within what we call a Sodarium styliferum. It's 11.3 in our 126 haplotypes of the supposedly to be Sodarium styliferum. This is much higher than the thresholds proposed by Herbert, for example, in 2003. He proposed that individuals differing in more than 2%, they were different species, and those differing less than 2%, they were the same species. So if we apply that, we are going to have many species here. And also, the diversification is really higher, much higher than some species pairs that we know that are different species. For example, the bonobo and the chimpanzees are species that there's strong consensus that we are talking about two different species of primates here. They diverge about 2 million years, and they have a CO1 like difference of 2.6. Humans and chimpanzees, we diverge about 13 million years, and we have a difference, genetic difference in CO1 of about 8.5, which is also much smaller than the diversity of the supposedly Solarian stelliferum. But yeah, sometimes I don't know if we are really the same or we diverged. So in case of the Bonetina, all the Bonetina species diverge more than 6%, so the 11.3 is really high genetic diversification within the genus. So let's go to the tree. This is the CO1 tree made of 126 Solarian stelliferum haplotypes. All the non-Solarian stelliferum were removed, but Solarian stelliferum forms a monophyletic group. So this is good, we have a monophyletic group. We don't know if it's one species or not, but they form a monophyletic group. And the support, of course, it's based on one gene, so it's variable. So the red branches represent branches not strongly supported, but we are not trying to assess the phylogeny here now. We are just going to assess the species delimitation. The interesting thing is that despite we had a lot of information and the differences between when you compare pair to pair using the hard gap barcoding method, the variation in the genetic distances is continuous between 0 and 3.2. Then there's a gap between 3.2 and 3.9, and then the variation continues until 11.3. So we have a barcode gap there that we were able to identify clearly. So we apply this barcode gap for the species delimitation. So specimens differing less than 3.2, we define that they were the same species. Specimens that differ more than 3.9, they were different species. If we do that, we get out of our Solarian stelliferum 13 species. So a lot of them. I didn't expect that variation, but it was that. Most of the species live in more than one locality, except these four here that they represent species supposed to live in only one locality, but most of them are in several. If we go to the phylogeny aware methods, PTP and GMYC, in PTP it's even more, we get even more species. It's about the same pattern that we obtain in hard gap barcoding, but two species of the hard gap barcoding. You can see here this red species and this one, the orange one. They are split, each of them in two species. That's why we have 15. And if we go to the GMYC, this was really messy. We got 24 here species, and it's not statistically significant, the result. So that result is futile. I mean, there's no information there. So if we go to the distribution of the potential species, despite there's a strong overlapping in the distributions, we can see patterns. For example, most of the species have a continuous distribution across the range. For example, you can see here central and southern Spain, avoiding the Sierra Nevada, which is an area with a high altitude and different climate. We have these other potential species here across northern Spain and Portugal, and central. And in some of them we have outliers. For example, this one has a distribution mostly here, and there's one outlier here. And also this one here and here. Remember that the Solarian stelliferum like the really disturbed areas, and they live under stones in these igloos. So the Iberian Peninsula have been populated by people for thousands of years, and people like to transport rocks. So it's not unlikely that some of these localities, some of these outliers are because of introduced populations from the human activity. There are a few potential species with discontinuous distributions, and 18 of the 74 localities have more than one potential lineage, which let's see what happens. So what's going to happen next? We still don't have the DDRA data. When we have it, we're going to use this data and the multi-locus delimitation to determine if these CO1 lineages really represent species or not, and to assess the utility of morphology and barcodes, therefore for species delimitation in the group and approach the phylogeographer. So, as a general and temporary conclusions, you can say that DNA is giving us a vast amount of new information to assess evolutionary processes at all levels. For species delimitation, DNA barcodes can be highly informative and a good step forward when compared with morphology-based delimitations. But DNA barcodes are highly dependent on the accuracy of the information of single markers, and different methods can give really disparate results. So, spiders of different groups and low dispersal capabilities show a high diversity hidden under similar morphologies. And this is to be a phenomenon widespread across neglected groups of invertebrates, including the megadiverse arachnids and insects. So finally, I would like to thank a lot of people that have helped me a lot across both projects. I'm not going to talk about the people in Mexico, I'm going to talk about Stano Pekar, which has been very helpful across the whole process, from going to the field to prepare the project to everything. Eva Lisnarova and Ondra Mihalek, they went to the field with me, they spent a whole week of their time working with me there. It was really tough because we traveled like 400 kilometers a day doing several localities. So thank you for this and for their support. Lenka Petrarkova and Lilia Servina have been very useful in the lab work and ordering stuff. Thank you very much for this. And support from the Masarykova University and the European Union, which are like funding this research. Thank you very much.
So thank you, ladies, for a very detailed overview of your past and the present. I would like to say that maybe this basically is almost beginning. It's not finishing, so I think you will be able to give us an updated version of the presentation of the new results later in next year. So we have time for questions. Yes?
I'd like to ask, you collected the tarantulas in the first part, and what are the methods of collecting those spiders?
Specifically the Bonetina, they live normally under stones. There are some tarantula species that they make burrows and some live under stones, that these species you normally can find it under stones, sometimes in like shallow crevices, but they really don't dig deep burrows as other tarantulas do. So in this point they are easier to find. But they are really strongly seasonal. So if you don't go in the right time, you are not going to see them.
How do you go about catching them?
I'm sorry?
How do you go about catching them?
Catching them?
Yeah, how do you catch them?
Okay, you use a flask and you just push them in.
So just take a bottle.
It's really easy. Yeah, they're really not aggressive at all. In fact, normally this group is not aggressive. There are many tarantulas in Mexico, they are not aggressive. When I compared them with the tarantulas in Cuba that I used to work before, it was a completely different change, because the Cuban tarantulas, they tend to bite all the time. They like it to make you suffer, but the Mexican tarantulas, they're really nice, so no big deal. And the venom is not strong. I mean, there's no tarantula in the world that has a venom that is really dangerous to people.
Maybe you could tell something more like what's the body size, if there is a sexual dimorphism in this species, in Bonetina, basically the whole genus.
So yes, the females they normally live under their stone. They tend not to move a lot, but males during the breeding season, there's a strong sexual dimorphism. Males have longer legs, and they tend to wander during the breeding season in search of females. But you can also find females. I mean, it was surprising that we found a lot of females walking as well. We don't know why, because this is not usual in other tarantulas. But yeah, basically, and you can also, I mean, males have all their life as juveniles. They have only one breeding season and they die after the breeding season. Just that. and there's a point in which they stop eating.
So do they build burrows and how do they hunt for prey? Do you know?
No, they just stay under the rock, and whatever comes around, they take it. Also, the males, when they are adult, they normally abandon the stone, but they don't. I mean, they can at the beginning still eat, but there's a point in which they just stop doing this.
Okay. Another question? Jirka?
You said that Zodarion would not use ballooning. What about the juveniles of your tarantulas hopping from island to island potentially to get to the north? Do they use ballooning as a dispersal method?
No, according as I remember, there's only one Megalomorph spider that use ballooning. They are
too
the
big, aren't
tenisids, small ones.
But even the juveniles are quite big, aren't they?
No, the juveniles can be really, really
Too
small.
tiny.
Can be really small. Yeah, the tenisids are like this, 2 centimeters. But the tarantulas, no, they are not known to do ballooning. So what about the dispersal across island hopping? So it could be rafts, for example. I don't know. But there are several groups that have low dispersal capabilities that share this. They were able to go from South America to North America before the rising of the Isthmus of Panama. So it's not really, I mean, the geological information is not really complete, so there could have been some kind of connection, but we don't know about it. Sometimes we think that the geologists have everything under control. They don't. So sometimes they don't have information about things that really influence. And based on the biogeographical information, sometimes we are able to determine backwards that some important events happen there.
Okay. Yes.
So are there some tarantula species or at least genera that can be found like in different parts of the world that are really widespread?
No. Tarantulas are they really live in they have restricted distributions. So most of the species are endemic to countries and small regions within countries. The diversification is really high, so no, and definitely not in different areas of the world. I mean, they really need normally a land connection to disperse from one place to the other one. And yeah, pretty much that.
Yes.
Are there any studies on spiders which have already compared species delimitation on single locus datasets and genomic datasets?
Can you repeat the question, please?
If there are any studies on spiders which have already compared single locus datasets on the species delimitation with genomic data?
Not that I'm aware of. Not that I'm aware. Yeah, because most genomic data have been used in spiders recently a lot, but mostly for phylogenetic reconstruction, not so much for species delimitation. So there might be something, but I don't know that.
I would like to say that it's a very new topic, and when I was attending last year, actually this year, a conference, so there was one single presentation that actually did it. There was a guy from Finland who has been comparing the multi-genetic data with the single genetic data, and he showed the differences. So it's a really new start on new studies. So any other question? Yes?
Could I ask you about genetic markers used in phylogenetic study? This MID1, MRPL, it's specific for spiders or terracosid spiders?
No, the thing is that there are very few markers available for spiders, so we had to develop several of these markers. So we did transcriptomes of 2 Bonetina species, and then we used all this database of transcriptome to try to find markers that were variable between the 2 species. And then we developed primers to sequence those markers. So like 3 out of the 5 nuclear markers, they are de novo developed during the project from transcriptomes.
Okay, another question?
Do you have an idea or an explanation for this morphological stasis in tarantulas? Given the age of the group, it's really surprising that it was diversified so much, and you mentioned they are not also specialized for a certain kind of prey like Zodarion are for why they do not change?
The thing is that they are not very diverse ecologically. So they are generalist predators, but at the same time they move all the time. I mean, there are some species that live in trees, but most of them live in the ground, in burrows or under stones. So they have not really diversified like other spider groups. And this is common to all megalomorph spiders. There are two big groups of spiders, megalomorphs and araneomorphs. And megalomorphs, they all the time share this kind of same habitat, same lifestyle, so they really have not diversified. So I really don't know if they have not diversified because their morphology hasn't been given them the opportunity to diversify, or all the way around, but the thing is that they are not really diversified in ecological terms or behavioral.
About the same group, what about sympatric occurrence and about competition between these species?
In
tarantulas generally, and in particular probably in your group of interest.
We are talking about tarantulas or Sodarium?
Tarantulas.
Okay, normally there were some localities in which there are sympatric species, but there are species like far in the phylogenetic tree. So in Bonetina we have basically two groups. One group of big species, colorful species. They live in really dry habitats, in strongly seasonal tropical forests. And there were other groups of spiders that they live in habitats with not so seasonal forests, higher altitude, higher humidity, but in some points they share habitat in some intermediate points. So normally the species that were sympatric were of this one of each group. So it's possible that there's and also they are different in size. The big species and the colorful are like this, and the small species they are really small. So I expect a niche diversification there.
Okay, thank you.
So, any other question? Yes?
Just following, do you have the data on the sympatric or parapatric speciation? We think they speciate alphatrically and they just came together, those species which are overlapping the distributions.
Given the fact that our sampling wasn't very deep, because 12 out of 17 species were known only from one locality, I cannot give you an answer. I have no idea. No idea.
Do they have aggregated distribution?
Yes.
Strongly aggregated, right?
Yeah. Strongly. Yeah.
Yes. So, Veronica?
In the first part you were talking about the different species delimitation methods and how they gave different results, and then you integrated them. So how this integration was achieved? Is it like computation or just by comparison with morphology?
What we did is that we tried to do this in a mathematical way to try to avoid subjectivity, because okay, we're trying to avoid the subjectivity in morphology, and we are going to do the same with the molecular. Now, what we did is that we implemented different setups for every method with the different possibilities, and there's no really a way to determine that this is a best setup than this other one. And we chose a species that were strongly supported as those that we obtained in at least 50% of the delimitations, but the same method with the different setups. So, and when we had the doubt if there was two species or one, we did a conservative solution, like lumping them as the same species, because you can have two errors, like oversplitting or lumping. So I think that it's best to avoid species and than describe a species that is not real. That's why we took the conservative way.
Maybe if I can also follow with another question. So you said that there were mostly some morphological apomorphies for these different species. So was it always the case, and what did you do when there was no difference whatsoever between the species, and what is generally your opinion about these molecularly based species without any differences?
In case of the Bonetina, we were lucky that 9 out of the 12 species used in the first delimitation work, they were easily to differentiate morphologically. So in the group of the big Bonedina species, those colorful, you normally find they're good characters. And in the other group, they're really more conservative, they're more similar. So after having the delimitation, we went back to the morphology and tried to find characters that define the groups. And we don't know because we didn't have a big series of specimens. Mostly we had from only one locality. So in the diagnosis, we said, okay, we found this, but it might be just an artifact of the sampling. We don't know. But we tried to give the diagnosis using the morphological features that we found afterwards, and also molecular data like distances, and also trying to find apomorphic changes in the sequences, the CO1 sequences.
Okay. Jirka?
What do you think about subspecies? It seems that molecular biology is kind of, I mean, people doing molecular phylogeny and taxonomics don't really like the concept of subspecies very much. Yeah. But on the other hand, it looks like quite a logical consequence when the split is not complete. So, I mean, if you're below this some threshold, you have obviously lineages which have parted some time ago. There might be still some limited flow of genes. This would be what we have traditionally described as subspecies, wouldn't it? So, how about that?
The thing is that I'm not sure about if there's species. So going to subspecies could be even a harder problem. So the different delimitation methods to determine species, they are really giving still different results and really disparate sometimes. So the concept of subspecies is not, I mean, if there's something, it's the species. You know, subspecies is something like we can use normally better for biological conservation or something like this, but I normally don't do it, and I try to not. I mean, I haven't get deep into this, but I think it's going to be a problem that it's more difficult than the species problem, and as we are talking about groups that they are not diverse still, they have not really diverse completely. We could, in theory, use them like when there's a conservation issues or something like this, but not as a general rule.
Okay.
Did you already sequence any Zodarian species to know the differences, you know, the amount of difference between species which are good described or defined by the morphology and other features?
So all the time we had like all of them from the Spain and Portugal. We had like 8 to 10 more Zodarian species, and the divergence between species was in the level of the 8 to 11-12%. So
pretty the same.
Pretty much the same. So we are talking about here that the differentiation between, I mean, the diversity within what we have as Zodarian stelliferum is 11.3, which is definitely very, very high. And then we have difference between morphological different species like 8-9%. So definitely something's happening there. The stelliferum.
Is there any other question? Yes.
From which part of the spider body did you isolate the DNA?
Okay, I started using one leg, but it was not enough. Yeah, they're very tiny and the concentrations were really low, so I finished, end up using all legs of the every specimen, and sometimes if there were really small portion of the carapace. I didn't want to use the abdomen because there you have gut content and you can get contamination with other things. Yeah.
It's a problem when if you isolate it from the whole body, it would be problem when you find in a sample the prey.
Yeah.
DNA of its prey.
Especially for DD rat, for the rat sequencing, because for CO1 I use specific primers that are not supposed to amplify anything else. I'm not using the general primers, but primers developed for spiders. But for the DD-RAT, the thing is that the technique is used like a restriction enzymes that they code the DNA every time they find a specific sequence. So you're going to amplify and read everything that is right there. So it's really like not a good idea to have contamination.
Okay.
The very last on my side. I just missed which method or data you use to calibrate the molecular clock for the tarantulas.
The molecular clock, we used the divergence between the Caribbean species that we included, like Formictopus from Cuba and also Cystophilus from Cuba, and South America. So the species of tarantulas of one of the groups of the Caribbean, they are strongly related with species from South America. This is something that it's supported also by for other groups, because there was supposed to be a connection between the Antilles, including Cuba, Puerto Rico, and Dominican Republic, with Northern South America about 30 million years ago, called Garlandia. And it is supposed that most of the biota in the island, they came through that bridge. Then Garlandia was split, but nowadays we have many groups in which this pattern is obtained. So we calibrated using mainly that. And also we use a fossil, not a fossil, but there was an analysis of whole megalomorph, like the infraorder, and the divergence of the Theraphosidae was calculated in about like 120 million years. So we use this as a top time of the diversification of the family. So basically we use on one side biogeography to calibrate, and also a previous phylogenetic analysis based on genomic data that has been published.
Did those match each other?
You don't know that because you don't do it separately. You use both calibrations at the same time and you get a result. But the thing is that if they don't make sense, so your analysis is not going to converge. I mean, if the calibration are really bad, so you are not going to have a good result. Like you are not going to have nothing because it's jumping all the time and you don't get stability.